The overall goal of this procedure is to analyze the gene function of mosquito larvae using recombinant mosquito Densovirus as a gene delivery.
This method can help answer key questions in viral-based delivery systems, such as how to overcome the associated extracellular and intracellular barriers. The main advantages of these techniques are easy manipulation, high transduction efficiency, long-term and robust lives of expressions, and the ability to produce persistent effects in vivo.
This work demonstrated that specific small RNAs or target genes can be overexpressed or knocked down in mosquito larvae using the developed Densovirus delivery system.
The implications of this technique extend the applications of the described expression strategy to investigate mosquito biology, because this method allows for easy transductions of larvae. Demonstrating the procedure will be Pei-Wen Liu, a doctoral candidate from a laboratory.
Start by ordering the chemical synthesis of full-length cDNA sequences for the precursor miRNAs, miRNA sponges, and shRNA as described in the accompanying text protocol. After receiving the ordered DNA, spin the tubes containing DNA blocks to ensure that all of the dried DNA is at the bottom of the tube. Then, resuspend the DNA in DNA.